What if you could erase a genetic disorder from your future children's DNA? Would you do it? This is the profound question at the heart of genetic engineering, a field that promises to revolutionize everything from medicine to agriculture. But with great power comes great responsibility, and the ethical dilemmas are as complex as the science itself. From curing diseases to potentially modifying human evolution, the stakes couldn't be higher. So let's dive into the fascinating world of genetic engineering. Genetic engineering has a rich history, rooted in the mid-20th century. Our story begins with a molecule, DNA, the very blueprint of life. It was first discovered in the late 19th century, but its significance wasn't fully appreciated until 1953, when James Watson and Francis Crick proposed the now iconic double helix structure. This discovery laid the foundation for modern molecular biology and inspired a new wave of scientific inquiry. Fast forward to the 60s, and the puzzle pieces began to fall into place. Scientists discovered the existence of enzymes called restriction endonucleases, which could cut DNA at very specific sequences. This discovery was akin to finding a pair of molecular scissors, and it set the stage for the next big leap in genetic engineering. In the early 70s, two brilliant minds, Stanley Cohen and Herbert Boyer, made a groundbreaking discovery. They developed the first recombinant DNA molecules, essentially cutting and pasting segments of DNA from one organism into another. This was an extraordinary moment in science, akin to the invention of the wheel in its potential impact. Suddenly, the genetic code was no longer a locked book, but a script that could be rewritten. This innovation sparked a revolution. It opened the door to a world of possibilities, from creating insulin-producing bacteria to genetically modified crops that could withstand harsh conditions. Scientists were not just reading the book of life, they were now authors, able to edit and rewrite the very fabric of life itself. But like all great stories, this one had its share of controversy. Questions about ethical boundaries and the potential risks of playing God began to surface. Yet, the promise of genetic engineering was too compelling to ignore. The potential to cure genetic diseases, increase agricultural yield, and even manipulate the genes of unborn children was on the horizon. This was just the beginning of an exciting journey into the unknown, a journey that would reshape our understanding of life and our place within it. Fast forward to the 21st century and we witness the advent of CRISPR. CRISPR, or clustered regularly interspaced short palindromic repeats, are sequences found within the DNA of microorganisms. These sequences are a crucial part of the immune system, allowing organisms to remember and defend against viruses. But it was the discovery of the CRISPR-Cas9 system that truly revolutionized the field of genetic engineering. Cas9, an enzyme that can cut DNA at specific locations, turned CRISPR from an interesting quirk of bacterial immunity into a powerful tool for editing genes. Like a pair of molecular scissors, Cas9 allows scientists to snip out problematic genes and replace them with healthy ones. The significance of this discovery cannot be overstated. Prior to CRISPR-Cas9, gene editing was a slow, expensive, and imprecise process. But with CRISPR, genetic engineering became faster, cheaper, and more accurate. It was as if we had moved from trying to sculpt marble with a sledgehammer to carving it with a scalpel, and the applications of this technology are vast. From treating genetic diseases to improving crop yields, to potentially engineering organisms that can help combat climate change, the possibilities are as limitless as our imagination. For instance, scientists have already used CRISPR to correct the mutation that causes sickle cell disease, a breakthrough that could herald a new era in medicine. But as with any powerful technology, CRISPR comes with its own set of challenges and ethical considerations. The ease with which we can now edit genes raises questions about how and when this technology should be used and who gets to decide. These are questions that we as a society will need to grapple with as we continue to explore the potential of this revolutionary tool. CRISPR has opened doors to possibilities that were once only dreams. As we stand on the precipice of this new era in genetic engineering, we are limited only by our understanding, our ethics, and our imagination. In today's world, genetic engineering is not just a concept, but a reality. This field of science has made leaps and bounds and now influences many aspects of our everyday lives. One of the most promising applications of genetic engineering is in the realm of medicine, with gene therapies leading the charge. By altering the genes within an individual's cells, scientists can potentially treat or even cure genetic disorders. Diseases like cystic fibrosis and sickle cell anemia that were once seen as inevitable life sentences are now being tackled head-on by the power of genetic engineering. However, this groundbreaking technology also raises ethical questions. How far should we go in manipulating our genetic code? Is it right to play God with our own nature? Genetically modified organisms, or GMOs, are another prevalent application of genetic engineering. These are organisms whose genetic material has been artificially manipulated to enhance certain characteristics, such as resistance to pests or improve nutritional content. While GMOs have the potential to resolve food security issues and reduce the use of harmful pesticides, they have also sparked debates about their safety and environmental impact. Lastly, we step into the controversial world of designer babies. This is where prospective parents could potentially pick and choose the genetic traits of their children. While the prospect of eradicating hereditary diseases is incredibly appealing, the ethical implications are immense. Are we on the verge of creating a society where children are designed to be perfect? And what does perfect even mean in this context? Each of these applications of genetic engineering carries with it a wealth of benefits, but also a plethora of controversies. The power to manipulate life at its most fundamental level is both awe-inspiring and somewhat terrifying. As we continue to push the boundaries of what is possible, we must also ensure that we are considering the potential consequences of our actions. Indeed, genetic engineering is shaping our present and will undoubtedly shape our future. As we gaze into the future, the potential of genetic engineering seems limitless. Let's imagine a world where genetic engineering is at the forefront of scientific innovation. One of the most exciting prospects is the potential to eradicate genetic diseases. Imagine a world where the likes of cystic fibrosis, Huntington's disease, and even some forms of cancer are a thing of the past. By isolating and manipulating the genes responsible for these conditions, scientists could quite literally cut out the problem at its source. But it's not just about fighting disease. Genetic engineering could also be the key to extending human lifespan. By understanding and manipulating the genes associated with aging, we could potentially slow down or even halt the aging process. Imagine living a healthy, active life well into your hundreds or even beyond. And then there are the possibilities of bioengineered organisms. We're talking about creating entirely new forms of life, designed to fulfill specific tasks. For example, bacteria that can clean up oil spills, or plants that can survive in the harshest climates and provide food for populations in need. Of course, with such power comes great responsibility. There are significant ethical and societal implications to consider. Who gets to decide what constitutes a disease or a desirable trait? What happens if these technologies fall into the wrong hands? And how do we ensure equitable access to these potentially life-changing treatments? These are questions we will need to grapple with as a society. But one thing is clear: genetic engineering has the potential to revolutionize our world in ways we can barely begin to imagine. Whether it's curing disease, extending life, or creating entirely new organisms, the possibilities are as exciting as they are daunting. Genetic engineering holds the key to many of humanity's most pressing challenges. As we move forward into this brave new world, we must tread carefully but also boldly. For in our DNA lies not just the story of who we are, but also the blueprint of who we could become. So, what have we learned about genetic engineering today? We've journeyed through the intriguing enigma that is genetic engineering, tracing its roots from its humble beginnings to its current cutting-edge developments. We've seen how the dawn of genetic engineering brought about a new era of scientific discovery, opening doors to insights into the very fabric of life itself. Then we delved into the rise of CRISPR, a revolutionary tool in our genetic toolkit. This game changer has allowed us to edit genes with unprecedented precision, making science fiction a tangible reality. It has not only accelerated research, but has also paved the way for practical applications in medicine, agriculture, and beyond. In today's world of genetic engineering, we've witnessed the impressive applications of these technologies, from creating genetically modified crops that can withstand harsh conditions to developing gene therapies that can potentially cure genetic disorders. The impact is far-reaching and profound. And the journey doesn't end here. We've also peeked into the future, a future filled with even more promise and potential. Imagine a world where we could eradicate inherited diseases, increase our lifespan, or even engineer organisms to produce biofuels. The possibilities are as vast as our imagination. In conclusion, the world of genetic engineering is a complex and controversial one indeed, but it's undeniable that it holds enormous potential for our species, promising a future where genetic disorders could become a thing of the past. Genetic engineering, while complex and controversial, holds enormous potential for our species. Before we go, remember to explore the world of science with us. Keep delving into the fascinating realm of genetic engineering, and don't stop there. The wonders of science are infinite, waiting to be discovered. To help spread this scientific literacy, consider subscribing, liking, and sharing our content. Your support makes it possible for us to continue sharing the wonders of science. Remember, the future is in our genes. Thank you for watching, and until next time, keep exploring.